Betsy, thank you very much, everybody. Good morning. Thank you for the invitation. It's a pleasure to be here today. And it's very interesting when Betsy reached out and we had a conversation about what it would be important for me to talk to the audience and having this varied audience and people from different type of industry and different parts of the actual local and state governments to listen to something that I can bring to the attention of how we at the agency and specifically at the Center for Food Safety and Applied Nutrition utilize the research, and that research help us to develop or provide guidance to the industry. So the next slide, please. So something very important is usually when I'm presenting, Betsy knows me, I have the tendency to provide a lot of information, and I'm always rushing and ending with the acknowledgement with just a flash. So today I decided that it was important to acknowledge all this team of people who actually made this presentation possible. A lot of the information I want to present today comes from researchers who spend a lot of time answering questions that we pose to them from the regulatory point of view, and that information they provide give us the confidence that the guidance or the regulations that we're creating are science-based. So first of all, I want to thank my management for believing that I can make a difference in this group. As Betsy indicated, I just recently moved to a position as a branch chief. So I'm directing the research that is being done to answer very specific questions regarding the safety of produce. The groups that I work with as the bacterial, genomic, and cell chemistry group, all these individuals, some of them already internationally known, work for the team that I lead. We have a group of experts in viruses. As we know, there's a lot of questions and concerns about the presence of viruses in food. Very important, the parasitology team, who has demonstrated that we can do things better, and definitely a lot of the questions that are being answered regarding Cyclospora cayetanensis are coming from this group. We have also something that I want to talk about is the Produce Safety Research Consortium, and we have two very valuable individuals who actually are helping us with this project. And last but not, of course, very, very important, my friend and colleague, Dr. Dave Ingram, that as soon as Betsy indicated what the topic of this presentation was going to be, I reached out to him because he has plenty of experience working with the industry and the regulatory aspect, and of course the research. So he and I talked, and this presentation is put together with his input as well. So thank you, Betsy. The next slide, please. So the next one. So it's something very, very important that we need to mention is that food safety, it's very, very important to know what's going on in our food. But also it's very important to know that when we recommend food to be consumed, from our point of view, we also need to consider that the reason why we're asking people to do is because of the nutrition value that they actually have. Thank you, Betsy. The next one, please. So when we see this is that, in the next one as well, when we have this conjunction of safety and nutrition, right, that's where we actually talk about health. So something very, very important that we need to all mention is that yes, our food is nutrition, but it also needs to be safe when it's consumed. And remember, very important, the role of the agency of my center is public health. So all these conversations I'm going to have with you right now is from the framework of public health. The next one is, please. So I just wanted to make everybody to be aware and to bring to speed that the agency, in this case the FDA, has authority over 80% of the food supply. So that's very, very important. We do have some share authority with other agencies. However, right, 80% belongs to the agency when we're talking about regulatory power over this. Next, Betsy, please. So, but something very, very important is that from that 80%, the ones that have represented more of the concerns that we have on produce safety or food safety are plant products and grain-based products. The next one, please. So that's something that we need to put on mind and think about when we're doing these type of activities. So, and as I mentioned, right, we have some dual regulatory responsibilities. But in this case, all these ones are the ones that we regulate at the agency. Next one, please. So, and now, right, to bring everybody even from the agency a little bit closer to where I am, is that I work for the Center for Food Safety and Applied Nutrition, right? Our mission is to make sure that that food supply is safe, sanitary, wholesome, and very important, honestly labeled, right? Also, something to make sure that people understand that Center for Food Safety and Applied Nutrition also regulates cosmetics products. However, this talk I'm going to provide today is not centered on that other side of the center. We're just going to be talking about food because that's where I have that background on, and I have a little bit of experience. So next one, please. So, and again, right, from that 80% of food that regulates at the agency, you know, CFSAN, right, that is the part of the agency who regulates or has the authority to work on food, is this something very, very important. For every 25 cents of a dollar that is spent, is an FDA regulated product, right? And that meaning that from those 25 cents that we use on FDA regulated products, remember, because we regulate more than just food, 75% of the 25 cents is spent on food. So for people to pay attention, you know, some people like to see how much we're talking about here based on money. And so if, you know, we have to put money on the, what we do at the center, CFSAN regulates $417 billion on domestic food and $49 billion on imported food. So it's something to put into perspective. All this is the work that we do at the center, and of course it's highly important from the public health point of view, right? And you know, something very, very important is that the American food industry accounts for 20% of the gross national product and employs approximately 14 million people in the United States. So we're talking about here domestically. So that's something that is very important to put into perspective, because all this will bring me to why we're talking about this. So next one slide, please. So that's what the center is. That's what we do. So I know you guys have heard about FSMA. I hope you have heard about FSMA, the Food Safety and Modernization Act, that it was became law in 2011. But a lot of people get a little bit confused, and they think this regulation, this law came out of nowhere. And just to put everybody into perspective, FSMA, as its name calls it, is modernization of an act. So we already had an act, a food act, Federal Food, Drug, and Cosmetic Act from 1938, right? So since that law was enacted, had been modified several times through the years for different reasons. However, this modernization of the Food Act was the one who gave us a different approach and made the groups that work on food safety and produce safety and public health, instead of becoming reactionary, be preventive. So that's a very important part. So FSMA modifies certain parts of the FD&C Act, but it's very important, certain parts of the Act. So anything that it was not modified by FSMA still stands at the FD&C Act. So eventually, we're thinking that based on the vocabulary that we utilize at the center, at the agency, and in the industry, although it is very important to recognize that FSMA was enacted into law in 2011 and modified the FD&C Act, still is the FD&C Act. And the vocabulary will be modified eventually to talk about the FD&C Act. And FSMA will just be remembered as the parts that of this change that modernized our food safety regulatory aspect in the United States. Next one, please. So why we do this? Unfortunately, right, the numbers, I know Mr. Morley indicated earlier, right, that there is a concern. There is a burden that happens based on foodborne illnesses, And then, the numbers, so these are current numbers from the CDC, right? So approximately every year, 3,000 U.S. citizens died related to foodborne illnesses that can be preventable. So that's why we are making sure that our modifications of regulations and all guidance reduces the likelihood that somebody consuming a food will develop an illness and or end up dying for the consumption of that food. So this is the objective that we have as regulators, and some of these regulations are basically being modified, and the guidance is being developed based on the scientific information that comes after we asking those basic questions. Why is this happening, right? So our reality is changing as well for the consumers. There's been some immunocompromised individuals that are becoming very susceptible to this type of illnesses. And we're talking about infants and children, right, because their immune system is not fully developed. Pregnant women who are with immune system compromised during their pregnancy. Older individuals whose immune system gets affected because of other situations going on with their body, age-related, and those individuals who are in chemotherapy as well, right? So you got to remember that some of these medications who help to control, eliminate, or modify the cancer also compromise the immune system. So for those individuals who also need to have a nutritious food, they need to have a health, safety food, and their health is paramount and is important based on the food that they're consuming. So, but we come back to the reality is that there's been some confusion that a foodborne illness is just a stomachache, and maybe a diarrhea, or maybe a headache, or maybe the chills. However, as time passes, more information we're capturing, and we're learning more about these conditions. We have associated some foodborne illnesses to other consequences on the health of the individuals who suffer of that foodborne illness. We're talking about reactive arthritis, where we have associated some salmonella infections who eventually have been linked to reactive arthritis. We heard earlier about kidney failure due to the consumption of O147H7 or STEC organisms present in food. And lately, we have been working with Guillain-Barré syndrome and associated to the consumption of Campylobacter contaminated food, so that eventually develops on this syndrome that is a very serious health consequence of having a foodborne illness. So let's make sure that when we're talking about this is what's going on. Thank you, Betsy. Yes, that's fine. But then based on that, right, so why did we the government of the United States, decided that we needed to have a modernization of the Food Act, right? Now, it's always based on a public perception, right? We expect as U.S. consumers to have high expectations on food safety. We expect the food that we're consuming is going to help us, right, through the health of our individuals, and it's not going to produce harm. So that's very, very important, and that's something that the FSMA had in mind when it was enacted. Also, right, we mentioned that adulteration and misbranded is very important, because we need to tell our consumer exactly what they're eating and where this food is coming from. And also, it's very important that we need to have the food supply constantly, right, available for consumers, and that disruptions do not cause a burden either on the manufacturer or in the consumer. And something that is very, very important is that when FSMA was implemented and regulations were created based on FSMA, the advantage that FSMA gave us was that now, before FSMA, we didn't have the authority over food produced outside of the United States, right? So not that we do now. However, the paradigm shift is because here now we have authority of the food that is consumed in the United States on its territories independently where it was produced. So that is very, very important because before we didn't have that authority, meaning that food now, it doesn't matter where it's produced. If it's consumed in the United States or its territories, then it must follow the same regulations as the food produced domestically. So that's very, very important, and that's something that it gives us now a better understanding that we have to protect our public of U.S. consumers independently where the food was produced. And of course, right, based on this, we follow the safe food trade, and we follow all our international requirements, right? The principles, very important, as we've heard this morning, is the responsibility of who is producing the food. They must be responsible for the food safety. So it is important for the producer of the food to follow food safety principles, and also, right, the accountability of the continuum from farm to table to make sure that everything they're doing is risk-based and the decisions they're making are based on their facts, and that make sure that they're practical and they're targeted, and meaning that the producer of the food needs to understand what they are producing and take into account where all these aspects during their activities may be of concern, and have a plan to avoid that this concern can be produce some human illnesses. So that's what is important. So now the producer of the food needs to understand in its totality where those concerns may be. And of course, something that we are doing is that the agency now is doing domestic inspections, and we, as I mentioned, have oversight of the import of the food that is consumed in the United States. So we have a mandate to do more inspections to assure and verify that the food that is produced and consumed in the United States is not going to produce harm to the consumer. And something very, very important that I have to mention is that we have strengthened our partnerships not only with the public, but with the private aspects and everybody who helps in food safety. So the agency realized that food safety is a responsibility of everybody, that we are working very close with industry and with academia, and we have created partnerships, and we continuously involve other players in food safety because it's important that food safety is a responsibility of everybody, and that's something that we believe at the agency, and we are constantly inviting our different partners to work together to answer questions and to develop proper communication so everybody understands where we are and where we're going on the aspect of public safety, produce safety. Next slide, please. And of course, and again, right, and all this is to that concept that produce safety was actually, that FSMA gave us what we call the 7 rules. I hope you have heard about them. You basically, I worked with the team that developed the standard for growing, harvesting, packing, and holding of produce for human consumption. That was before the job that I have now. And that is what's called the produce safety rule. And I was very lucky to work with the teams who developed some of the materials for the preventive control for human foods, or the PC rule. I've taken classes on the other. We all need to understand the other regulations that came out of FSMA, but we're going to concentrate on the produce safety rule just because that's something that I have experienced as a researcher as a regulator and as a researcher now again. So next one, please. Let's see. Yeah, thank you very much. And something that a lot of that we don't talk enough about is that the produce safety rule needed some data to be to be cemented in science. So there was a team of researchers who developed a qualitative assessment of risk that indicated why a produce safety rule is important and where we should span our concerns or ask those questions that are relevant to make sure that the food that we're producing is safe. So this is very, very important that people do understand that the produce safety rule was cemented on a risk assessment or assessment of risk, where we ask several questions. The next one, please. So, and the question had to be relevant to the objective that we had. And always, that I've always have indicated to the teams that I work with is that the information we're going to capture is going to be tied to the question that we ask. If the information that we're capturing doesn't answer the question, is because we asked the wrong question. So when we at the agency were developing this assessment of risk, those are the questions that we had on mind or we asked to be able to cement our produce safety rule in scientific data. The questions, the basic questions are: what are biological hazards of concern? How does produce become contaminated? What is the likelihood of that contamination that vary among the different types of commodities that we are producing, right? And remember, this is the produce safety rule. And something is what is the likelihood of illnesses attributable to produce consumption. So it's not only that the produce is contaminated, but also the likelihood of that linking that consumption with the illness. Also, practices, what is the impact of post-harvest practices, right, to the contamination level? What are we doing that may reduce or increase the likelihood of contamination? And what is the activities that a farm is doing to reduce the likelihood of contamination? So those are the questions that we asked to develop the produce safety rule. And we had to answer those questions based on the data that we had, knowing not only at the agency, but also in the United States and outside of the United States as well. The next one, please. So the answers that we obtained was that we actually identified that the microbial hazards are a significant risk and a recurring risk. So that was something very important that we responded. A lot of people think, well, that was known. But yeah, now we have the data to prove that that's the reason why the produce safety rule is science-based, is because we were able to identify that effectively microbial hazards are significant. During the same answering of those questions, we did a hazard characterization, and this is the list of microorganisms or pathogens of concern that we have identified as the culprit of the majority of the illnesses associated with the consumption of produce in the United States. Now, they're not in an order of importance. They just, they are there for you guys to know, actually almost alphabetically, just with the exception of the parasites at the bottom. And that's something that it was important for us to know, where are we going to develop regulations and guidance for our industry to pay attention to. And it's something very, very important that we also actually identified is exposure assessment. And we identified that it's very important to identify any source of contamination, but not only the sources, but the routes of contamination, and linking them together. That's where we can answer questions about what happened when there is an illness associated. So we need to know the source and the route, and also what is the likelihood of the contamination to occur during growing, harvest, and post-harvest. And that's why the name of the regulation includes all those activities, right? And then what is the likelihood of an individual consuming that product and, of course, developing the disease. So, and of course, we did a risk characterization, right? And that's what came out as the produce safety rule. The next one, please, Betsy. And of course, as I mentioned, right, something that we identified is that all these situations, all these activities, characterizations, or points do introduce pathogens on produce, or domesticated and wild animals, and of course the birds. Can be domesticated wild, but there's actually a very, very important. They do introduce pathogens on produce, the environment, and certain weather conditions such as floods or intensive rains, or pieces of equipment that we're utilizing during the harvest or post-harvest activities, or workers who are in constant contact with the produce, either during pre-harvest, harvest, or post-harvest. The irrigation waters that is highly important in something that we talked a lot, and that's highly important is our soil amendments, and that's something that we know, right? That if the soil amendment may be a potential source of contamination. So that's where we need to center our discussions and our learning and our regulation and our guidance, right, to talk to the industry. So the next slide, you just click in and you see, so those are the areas that we need to pay attention to when we are asking these questions, where in my process I may have a concern and can I do something about it? So the next slide, please. So as we were developing the guidance, and we had more questions than answers in some parts, right? That doesn't mean that we're not going to do something about it, right? Having more questions is good because give us the opportunity to ask more questions and create more data and have a sound-based regulation and guidance. And something that we keep on asking ourselves till this day is, what is the origin of the pathogen in the farm environment? Where is the human pathogen coming from? Remember, we're talking about Escherichia coli or 147H7, we're talking about Listeria monocytogenes, we're talking about Shigella species, or Salmonella, or the parasites, right? Even norovirus or Hep A. So where are they coming from? Where are those sources of those pathogens in the farm environment? So that's something that we still are asking those questions and constantly getting data to answer that question. But it's not only that it's there. How does it survive? How long does it survive? How does it move to the farm environment? Who moved that pathogen, right? We hear about that there is a source, but what is that route? How does the pathogen end up in our produce, right? And are we talking about animals moving it? Are we talking about the soil being the one who help us to be the pathogen to stay longer? Is it the water a vehicle that we have to be of concern? Or what activities are we doing that actually are affecting the presence of the pathogen, either eliminating or enhancing the presence of the pathogen? So we are constantly asking these questions. We're constantly generating data, and that's something that we need to understand. Very, very important. And then, right, as I mentioned, what are the sources? How it end up in the produce? How is it transferred from all those sources? What is the vehicle of transferring the pathogen into the produce? How does it survive, right? Again, does it survive in produce, right? It's not only on the environment, right? But once it stays in the produce, how long does it stay there? Is it going to produce an illness because it's in produce, right? On the produce. And you know, very, very important, and something that is important and critical for everybody to understand, the produce safety rule is based on practices. There's no inherited reason why a produce is going to be contaminated. It is the practices associated with the production of that specific produce, the ones who may be the reason why a produce ends up contaminated. So highly important to pay attention to our practices. What are we doing and what can we improve? And of course, right, it's not only that the produce is contaminated, but it's in levels of that can produce the illness, right? And you know, some we are able to identify with some pathogens of concern that the numbers that we may need or may be present in food that may develop a disease, but we still have questions or pathogens of concern that we specifically don't know the virulence of that pathogen specifically. How many cells do we need, or not that we need, but are they present in the food that may develop the disease in the person who's consuming that specific produce? So those questions, we keep on asking them constantly at the center to make sure that we provide proper guidance to our industry. Next slide, please. So, and again, right, one of the parts that it was not closed during the produce safety rule is subpart F, that is biological soil amendments of animal origin. We knew when we were developing our guidance, our regulation, that we needed more information regarding biological soil amendments of animal origin. And we decided to create a risk assessment. We didn't want to postpone the rule until we finalized the risk assessment. We decided to actually close the rule as is right now, made it final, with the understanding that we were going to eventually do rulemaking to address the questions regarding the application and what the questions that we asked originally on our assessment of risk regarding biological soil amendment of animal origin will actually provide a more robust regulation. So we got in the task to capture data and to analyze the data and to make sure that we provided proper guidance to our industry about the application of those soil amendments of animal origin that are utilized in the production of fresh fruits and vegetables. So that was something that it was created to capture the data, and we are in the process of analyzing the data and writing a proposed subpart F to get comments from the industry based on the information that we had captured and what we have observed in not only how we utilize those soil amendments, but the intervals of application from those soil amendments when they are not modified to be composted, right? So that's something that is very, very important for people to know, that we are working constantly on that and capturing data and answering questions. So the next slide, please. And so again, we're going back into what's happening. I know during the talk earlier, we've seen that the presence of domesticated animals may have an influence on the presence of pathogens on produce, right? And we know for a fact that soil amendments of animal origin may have pathogens when they are obtained, right? So animal manure may potentially have human pathogens. So that's something that we know, and we need to know how we apply these soil amendments into our soils and or before, during we are growing our produce. Next one, please. So the next questions that we need to ask is, well, now that we know that we may have some pathogens, how long do they last in the amended soil? Do they actually stay there forever? Do they die off? Do they get affected by other competing organisms? What factors are important to determine that a pathogen may not survive or may survive longer? Are we talking about the presence of certain nutrients in our soil? But also is very important soil moisture, the availability of water for our organisms to survive, and what is the temperature, right, of the soil during the time that our pathogen survives. The next one, please. So then, right, it's not only to find out that there may be pathogens that may survive, but how are they transferred from the amendment to, you know, the actual produce of that we're going to be consuming, right? And there's a lot of data being generated about splashing water, right? If the pathogen was in our soil and we have an activity that splashes the water from the soil in, you know, to the plant of concern or the produce, what is the transfer of the pathogen from that soil that potentially was contaminated because of the application of soil amendments, and then it's you know, it ended up on the produce, the same as the wind. That the wind has a factor in moving those pathogens as an aerosol. Are the pathogens surviving and making possible they can be aerosolized and move from one location to the other because they were on the soil, because they were applied with, you know, as a soil amendment, right? The pathogens were in the soil amendment, and the wind is carrying that. And of course, you know, the presence of animals, right? That is as I mentioned earlier, the presence of domesticated or wild animals is, you know, when they, you know, defecate in the fields, there's something going on there too that would make the pathogen of concern more protective of the environment, because they stay on the feces as feces, right? And they're not composted, or they're not incorporated, or they're not, you know, modified in one way or another to become part of the soil. The next one, please. And then, of course, right, what are we doing in our fields that are going to impact the presence of the pathogen, right? The idea of modifying of harvesting practices may have a concern, because we may be transferring the pathogens, if they were on our soil, into our produce. And as simple as leaving an unprotected produce in the soil and then bringing it back into to be consumed by our individual, right? Did we transfer pathogens from the soil that, if we hadn't put that produce on the soil, wouldn't have transferred the pathogen at all? So, and there's a lot of questions that we're still asking, but it all has to do with the soil amendment of animal origin, right? So application of that soil amendment and everything that we do till the final products are the questions that we're asking and trying to answer based on this risk assessment. The next one, please. So with that, right, so again, it's important that soil, right, it needs to be healthy to produce healthy food. So next one, please. So, but we cannot just think about soil and as an isolated entity. We need to be aware that because of the nature of the industry, the majority of farms in the United States utilize soil as a substrate to grow the plant. We need to make sure that the food safety is an important component when we are thinking about the soil that we're utilizing, right? So it's not only about the microbial presence, but also the chemicals that may be present of the soil. The next one, please. That, of course, is going to give us our consumer confidence that we indicated earlier. Our consumers expect to have food that is not going to produce any illness. So when we as a farmer, as the industry, think about growing our produce, we have to consider all these points and all these questions. And this is something very, very important. The next one too is because everything that we consume make us better, right? So that's something that we need to consider and think about constantly as a producer. What are we doing when we grow to make sure that the benefits of consuming what we're growing is a good product that is going to enhance the health of the consumer. Next one, please. So that's the part of the risk assessment that we are developing at the agency to answer all the questions as to Part F. But something that I wanted to talk to you about also is what I'm doing right now as working for the Office of Applied Research and Safety Assessment. When I was hired to be part of this great office, I heard about this Produce Safety Research Consortium that the office had already created. It is a very interesting project that it was organically developed. So it was the University of Southern University of the United States reaching out to the federal government, or the federal government reaching out to certain universities, just because we were interested on working with them because of the projects they were working on already. So that's how this consortium was created. It was one by one the universities that are working with us, or our sister agency like USDA, is because of the projects they're working on. So the consortium is already leveraging of ongoing projects. So that was, we did not came out with a pot of money that we don't have, and we didn't give it to university to say, hey, do whatever you want. It was like we were heard about, oh, you're working on this project, that sounds very interesting. We have experts that can help you to answer some of the questions, and you already are developing research to answer some of the questions. Let's work together, right? So based on the leverage, we are facilitating collaborations and answering some of those questions that we have to fill those gaps, not only for the produce safety rule, but also for guidance and other questions regarding different regulations that we are actually working on. Something that is very important is that we provide produce related on-farm environmental collections, meaning that we are working with universities and farmers to identify research areas of concern, right? So that's something very, very important that we wanted to make sure that we had. Of course, we are supporting not only the produce safety rule, but other FSMA regulations. And something that is highly important, and we're expanding our contributions to the genome tracker, and I'm going to talk to you guys about that in a minute. So next one, please. So the objectives, very general, I just wanted to put them there. I know you're going to be having access to this presentation. It's being recorded, but it is important to identify different practices done in the United States and abroad. We're working with partners of Canada and Mexico, or universities in the United States who have partners in Canada and Mexico, to see what practices are being different and what is the presence of certain pathogens of concerns on those soils, right, who are producing the food that we consume in the United States, right? So not only is about supporting the produce safety rule based on our pathogens of concerns, is also identifying those practices and finding out what practices have an influence on on the presence of pathogens on the product at the end. So something very important is that when we work with the universities, is the university the one who is working with the industry. The industry and the university work together. The university collects samples of soil, water, and they isolate if any pathogens of concern, and they send us the isolates. The university identify the presence of pathogens in produce, but we do not have that information. That information is kept by the university, and the university talks directly with the industry, in this case the farmers, to provide the guidance that they need and the education that they need to make sure that the livelihood of a pathogen ending up on produce is diminished. And I'm using this information that is critical and important because we want to make sure that the industry continue trusting the university partners, And the information that is sent from the university partners to us is just to see what is the reality of the farming community in the United States without having the information that there is concern, right? Because the industry is being guided by the universities to make sure that no concerns arise for the consumption of the produce that they are growing, right? So the next one, please. So we are asking all these questions to ourselves. We want to know what is the microbiome of these soils, water, produce, and we want to find out if there is any antimicrobial resistant genes on the pathogens that the universities are identifying, right? So we do whole genome sequencing of the isolates the university sends us, right? And that helps us to understand more the reality of the farming environment, right? What are the areas of concern? And we capture all the information that is important to identify where our guidance needs to be created, right? Are we using amendments, as I mentioned earlier, and how they're being used, right? And very important is the presence of animals in your farm environment a significant contributor of the presence of pathogens? And if so, what can we do to reduce the risks associated with the presence of these animals, because they may be a source of pathogens? So again, all this is in an educational and outreach component, and we're working very close with the universities. Once the university creates this data, then we can help us to identify areas that we can actually create better guidance based on the data that are being captured. The next one, please. So, and what I wanted to also indicate is that we work with these universities, and what we provide is DNA sequencing so they can actually do their analysis if necessary and try to identify any trends or tendencies to the presence of pathogens, or if the pathogens are really surviving in the environment. So that information is created, but is given back to the universities to make sure that they have all this information and they can create these communication documents within the industry and improve, right, the industry and reduce the likelihood of any potential contamination. The next one, please. So again, right, so we talked about soil properties, we talked about microbiome, the presence of organisms in the environment, right, the application of soil amendments, but something that we are thinking lately a lot is, well, how about air? You know, we mentioned that the air may be a potential route of contamination, but we're not sure yet. So we're asking ourselves those questions as well, and we're trying to see, can we answer this question, right? So something that we need to make sure is that we answer those questions, but that has to do, what question are we asking? Do we have to include any concerns about these pathogens being aerosolized and moved from a source as being the air as a vehicle and end up in our produce? So next one, please. So that's something that we are asking ourselves. And I just wanted to let you know that we work very close with Ohio University of Ohio, University of Georgia, and we have already captured data, and we have analyzed some of the data from 3 years of communicating with them and having this sample being taken by our partners. So the next one, and something that we started to see was the tendency, right? So something that's critical is that the soil type and the time of collection of those soils of soil that identify a different microbiome. So you can see on your slide on the left side, you can see that composted soil have more microbiome. There's more organism presence that soil that is not composted, adding compost to the soil is not amended. And then you can see that the variation also has to do with what time of the year we actually are collecting those samples. So that's something that is going to help us to create more guidance for our industry and start discussing with them when you're applying your compost and how long this compost is going to last for the capabilities of the soil to be healthy, but at the same time that if there's any potential of pathogen of concern, how can we apply this and for how long can we apply it? Again, we don't want to just give numbers. We want to make sure that our numbers have a significance and is based on the capture data that we're generating right now and all the analysis that we're doing. Next one, please. So something also that is very important that we're working on is that, you know, it was the case mentioned about the presence of 0157H7 on lettuce, right? That we know the case of 2006. But something that we are asking ourselves is, and we're working very close with USDA, is that what happens when 0157H7 is present in fresh cut lettuce and we apply modified atmosphere? So that's something that we had this question ourselves, and we work with our partners with USDA and try to answer this question. So our partners in USDA grew the lettuce, right? They grew 2 different types of lettuce. They actually did do some processing, meaning they washed it and they cut it, and then they actually inoculated the lettuce and they put it in modified atmosphere packaging to see what would happen to the presence of 0157. The next slide, please. So we did metagenomic analysis, and we identified that actually some the pathogens of concern do have the presence of the microbiome moves from depending on where we actually are affecting or testing for the presence of pathogens, right? So we have, you can see on your graph, this and this and the colors. The most important part here is there were 2 different varieties of lettuce, one with good shelf life, one with poor shelf life. And if you can tell here, the microbiome shifted from unprocessed lettuce to inoculated processed lettuce. So the addition of a foreign organism shifted already the environment of the presence of pathogens. And although you guys cannot see here, and I highlighted in a little blue square, there is actually some of the presence of 0157H7 there, right? But it was inoculated, so it was not present on the lettuce, right? It was inoculated, but then after 6 to 14, after storage of 14 days, that presence really decreases. So we're asking us questions. What does it really mean? Of course, we don't want to have 0157H7 on our food at all, but we wanted to see the effect of modified atmosphere on the presence of this pathogen, right? So the next slide. So that's some of the questions that we're asking ourselves and working that. So almost to the end. So we have a data. We're working for 3 years with this consortium. Not all the data that we have is analyzed. Unfortunately, our 2019 data started coming in January and February, and due to the current pandemic, we were not able to do all the analysis that we wanted to do at the beginning of this calendar year. But based on these activities working with our partners, we realized that we needed to develop new methods for the detection of other pathogens of concern, such as viruses, such as Socrates, and other bacterial pathogens, such as Campylobacter, right? And we realized how important the role of metagenomics is to find out the environment that these pathogens are under, right? The conditions, and how these metagenomics, the presence of these other organisms, affect the presence of the pathogens of concern. Next slide, please. Now, future, right? So this is where I'm talking to you about the 3 years. So of course, we're going to continue analyzing this data. We are expanding our consortium. We have invited universities that belong to the Northeast Center for the Advancement of Food Safety, NYSAFP, that is in the Northeast. We're working very closely with other universities with the North Central and Southern regions of the United States as well, for them to be part of this consortium and to create more, a better pool of data. We are right now in the process of establishing what we are calling a growth chambers and growing rooms research facility here at my office, where we will be able to simulate real-life environmental conditions to answer some of these questions, some of these gaps that we have about how the pathogens survive and transfer. Because these are BSL-2 labs, we can utilize full pathogens. We're not going to utilize attenuated pathogens or surrogates. And we want to see how all these factors affect the transfer and survival of pathogens, and also the soil types, because as I mentioned, some of the importance, not only the humidity and the temperature, but also the type of the soil for the survival of the pathogens. Specifically with pathogens, we are trying to answer questions regarding the sporulation of those pathogens and the survival of these pathogens, in this case, Cyclospora cayetanensis, and some of the projects that we are involved in right now. So the next slide, please. And I believe this is my last slide. We are actually simulating some studies through the years in this applied growth facility. We have the background of working with the industry from the Delmarva Peninsula, Delaware, Maryland, and Virginia for the presence of Salmonella in tomatoes. That was a very successful partnership that we had, and we want to replicate that in our growth room, where we're going to be able to control the temperature and the humidity and go down to 28 degrees Fahrenheit and all the way up to 95 degrees Fahrenheit and see what happens in an entire season with the survival of the pathogen, if there's pathogen in the soil. And so it's, again, all the data that we are creating here is not only to just for the sake of science, but all the data that we're creating is to help our partners and not only the industry, but only the academia, and to make sure that we have better, stronger, and more substantiated guidance based on science. Suggest, create data for our industry and our universities to provide guidance on growing practices. And something very, very important is the usage of water. That is always very concerning, and that is something that we continually working to improve. The next one, please. And thank you very much. That's it.